So it's my pleasure to introduce David Glann, who's replacing John Berngero, who couldn't come, but we have a fantastic replacement. So thank you, David, for coming up.
Good morning, everyone. This morning's talk is focusing on rare variant discovery with extended pedigrees, and we'll focus on some data looking at human subcortical volumes. As JB mentioned, I am not actually the person you signed up to see, who's John Berngero. He's slightly taller, much heavier, and with much more hair than me, and is often much more enjoyable. Occasionally I will try to channel Dr. Berngero as I'm making some of these points, and when I do, I will probably do things like bang my fists on the lectern or something along those lines, and then you'll know that it's not really me, it's actually John standing here giving the talk. Okay, I guess I would say that, which of these buttons am I supposed to push?
This one is only for the slides.
Oh, Mac. Oh, got it. Oh, much.
Slides go,
yeah.
go this over your
There we go. So that's me. David Glann. I'm at Yale. Lovely. I don't have any conflicts of interest. Okay. So where do we want to start with this? And I thought I would start by kind of anchoring us to this slide that we've seen once before today. And this is from Stein's first Enigma GWAS paper published in 2002, looking at hippocampal volume and at intracranial volume. And really what happened here is there were about 20,000 or 21,000 or so subjects. We went in, we looked at common variants influencing hippocampus and common variants influencing in intracranial volume. To be sure, this was a meta-analysis, and Sarah was exceptionally involved in doing that. So a lot of the people you've seen here today were involved in this particular study. And this, I believe, was a really important study because it was one of the first times that we were actually able to do a large-scale GWAS with an imaging trait. More recently, Derek's paper came out earlier this year, where we then upped the sample size closer to 30,000 individuals, and we went in and looked at a bunch of different subcortical regions. Here, particularly exciting was a big hit we got for the putamen. But nonetheless, we are kind of replicating and extending the hippocampal findings. Things seem to be working reasonably well within this common variant meta-analytic strategy. But then we begin to think about, well, what exactly are our goals here with imaging genetics, and really what are we doing? So when you think about the genetics analysis of complex traits, there are really kind of three basic things that you want to do. You want to do a QTL localization, and that is, where in the genome is there a quantitative trait locus influencing the trait that you're interested in? That's the first level of search you want to do. The second is you want to go from QTL localization to gene identification. So now that I have a QTL, where is there a gene or multiple genes that are influencing that trait present? A QTL localization does not give you a gene identification, and we'll talk about what the difference between that would be. And then once you are lucky enough to actually understand and think that you truly have a gene present, you would then go into QTL, the QTL allelic architecture. So in this case, you would go in and start looking at, well, what are the SNPs? What are the variants? How are they influencing the trait? In this case, we use the term QTN, which is quantitative trait nucleotide, but that's just for fun. And then we go in and we look at the frequencies of effects, and you start to look at different ways in which the allelic architecture might allow you to predict incoming new cases, how they would or would not look based on these data. So what's the status of QTL localization? So because of meta-analysis and GWAS analyses and some family-based linkage studies, we are accumulating a very large number of QTL localizations. As a matter of fact, pretty much all of the things that you get from GWAS is giving you a localization. But that localization does not give you the gene, per se. It's giving you a localization. And then part of that has to do with like things that were talked about before, doing with LD structure, or that the variant that you're finding isn't actually the functional variant that you think is relevant. It's going to be somewhere near that. So if you're doing something like family-based linkage studies, your QTL localization region could be huge, right, between 10 and 15 megabases. So there could be literally thousands of possible genes that could be the culprit associated with why you're getting this effect. If you're doing association-based measures, and particularly chip association-based measures, as we were discussing before, and depending on what the local linkage disequilibrium structure is, you could be looking at about 500 kilobases, so about 250 kilobases on either side of the variant that you're interested in. There could still be hundreds of possible genes within that. And so we have to kind of get past this notion that, oh, well, we got a genome-wide significant SNP, and that's telling us exactly what the gene is. Well, you actually need to do more work to get there. And the identification of the underlying genes requires a number of different things. One thing that most people tend to agree on is that it requires deep and comprehensive sequencing of the regions to find the variant, the functional variants. Typically, people believe that that has to be done in the exact same sample that you've collected. So now we have a problem, because we have a problem that we've moved to doing meta-analysis, and people aren't actually sharing the data, particularly the genetic data that's present. And so we have to figure out ways of coming around and dealing with this. And then you start to think about this, and you start to think, well, if the SNP array that we're using, which has 1 million SNPs on it, isn't actually giving us enough data for us to make a definitive statement about a particular gene, right? And I will point out here that I actually believe that you need biological confirmation before you're going to get to a gene, and I believe that's what Jason Stein will be talking about in the last talk. But you start to think about it. Well, this is a daunting problem, because the genetic search space is huge, right? But there is one thing that's advantageous to it, which is that it's huge, but it's also finite. Right? So we have 23 chromosomes, somewhere between 20 and 25,000 genes, some possible 3 billion possible variant locations. Okay, that's a big space, but it's also it's a finite space. You can compare that actually to the phenotype space, which might actually be infinite. Right? My genetics colleagues laugh that in imaging genomics I can come up with 25 to 40 phenotypes for every single thing that we think we're going to measure, right? Because you can measure shape or volume or whatever. And so that might be an infinite space. So we do, however, want to focus in on as many of those possible variants as we can, and that might push us to whole genome sequence, as was discussed in the first talk today, which is really getting in to say, okay, what are all the possible variants that we could be looking at? What's important here to note is the number of variants you have for whole genome sequence actually scales with your sample size. Because if a variant, if a locus is not variant within your population, you say that it's invariant, right? But that might be the fact that you only have 1,000 people. If you had 25,000 people, it might actually be that some people might have variation within that locus, and therefore it expands your size. Okay, so if we're thinking about that, sometime this will go, clicky, clicky. Okay, so I was thinking about this on the flight here. So this might not make any sense at all, but this is what I was thinking. Okay, we know that there are about 67 or 6.77 billion people on Earth. Okay, we know that the estimate currently is that each person has 80 new mutations, approximately, right? So we all have about 80 new mutations. If you multiply those two, that gives you 536 billion new human mutations on Earth at this present time. Okay, that's a lot of new mutations that are present. And if you think about that further, you would say, okay, well, if you divide that by the total number of locations that you think are present, you have 162 possible variants per each site in the human genome, which is huge. Of course, that's not actually true, because some sites in the human genome are going to be incompatible with life. So there are some variations. But what I'm saying to you is the potential for us to find rare variation and variation by each locus in the genome is really substantial. And we don't really tend to think about that when we tend to do this type of study. But when you start to do human genome sequencing, you do start to think about that, because you start to think about, well, what's the probability that there's going to be something unusual here? Okay. I wonder where I'm supposed to point this. Oh, there we go. Okay, so there is a growing realization from whole genome sequence studies that the vast amount of genetic variation in the genome is rare, right? Or possibly even private. And that sequence, that common variation that we primarily look at and have thus far really accounts for only a small proportion of the total observed heritability of a given trait. So why is this? Well, because here's some data from my own lab, but it's very similar to data from the 1000 Genome Project, where what we did is we took whole genome sequence data from about 600 people in this case, and we bin them by the minor allele frequency for each of the variants that was present, and then we did a proportion. We showed that about 67% of the variants that we were able to identify had a minor allele frequency of less than 0.01%. So that's telling you that a substantial amount of what we now consider to be human variation is rare. And I would say that there's increasing evidence that rare functional variants contribute to complex traits. So if that's the case, and we do begin to say, okay, well, if the vast majority of the variation is rare, and we think it's important for the kinds of traits that we're interested in, how do we study it? I'm going to skip a slide at some point, and then I'm not going to know what I'm talking about, but it's okay. Okay, if a substantial component of quantitative trait variance is due to rare sequence variants in multiple genes, then we could have a hypothesis, primarily the rare variant hypothesis, which is now we want to go and search for these kinds of more rare variants. And then if that's true, we now have a power problem, right? So if we had a power problem and we were only going to search 500,000 variants or 1 million variants, we're certainly going to have a power problem when the total number of variants that we're going to search becomes much larger than that. So we end up kind of coming to a compromise already, where we say, okay, larger effects of rare variants will make it gene discovery easier. So it's not that all rare variants have large effects. It's that the only ones that we're really going to have the ability to see are those large variants that have those rare variants that have large effects. And it's going to be easier to perform the required functional experiments on variants with large mean displacements. And what do I mean by that? Well, I created, whoopsie. I'm sorry. I don't really know where I'm supposed to point this thing to work. Towards you? Ah, very good. Okay, QTL effects. So this is a heuristic table that I've been kind of working on for a while to let us think about what it is that we're looking for. So in a rare monogenic disease, we know that a particular variant is going to have 100% of the heritability of the illness. We know that this is the variant that's going to be there. We expect that the difference between the person without this variant and the person with the illness will have a substantial displacement between the two of them and their genotypic means, meaning a substantial displacement with relationship to the phenotype. And if we think about this in standard deviation units, we would think that globally people are estimating that a rare monogenic disorder is giving you a difference in the phenotype of greater than 3.5 standard deviations. So that's a whopper of an effect, right? So realize that we're recalculating how we're talking about the impact of a particular genotype from thinking about it of relative risk or of very small effects that we might see in common diseases to trying to say, okay, we want to see big effects. In contrast, if you look then, for example, at a GWAS derived common variant, there it's usually the frequencies are greater than 0.01%, which is kind of what Sarah had mentioned before, the things that you throw out when you're doing this kind of thing. And there, in general, we're seeing displacements. If we get a good effect, it's about 0.15 standard deviation units. Right? So it's really relatively small, which is exactly what we're seeing in GWAS studies, not just within imaging genomics, but in general. And when you're looking for the rare partially penetrant models, what you're looking for are heritabilities that for a particular location that are due to 0.1 or greater in a given pedigree, and that you're expecting to see an effect of between about half a standard deviation to about 3.5 standard deviations. Okay, so you're expecting to see these kinds of large effects, and that's really what we're searching for when we go to look at this. So how can we study rare variants? So at this point, John Blangiero would bang his hands on the lectern like this, and he would say, you need to study families. And then that's really going to be the most channeling of John I'm going to do today, except maybe later if you stop by and we'll have a beer. But at this point, really what we're thinking is rare functional variants are best detected and their genes identified using large pedigree-based designs. Why? Because pedigrees observe the observation of multiple copies of a private variant. It's really simple. It's basic Mendelian mechanics, right? Large pedigrees with large lineages are optimal for studying private variants because the potential to generate sufficient numbers of private allele copies exists. So how do we think about that? Well, what this is telling you is if you have something rare and you're studying individuals who are related, the likelihood that you find multiple copies of that rare variant increase. If that's the case, you have better power to say what that rare variant happens to be doing, right? So, so how would we think about that? Here's some data from one particular family in a study that we've been doing. This is the San Antonio family study, and these were data that were sequenced as part of the T2D genes consortium, so the type 2 diabetes consortium. And what we did is we just looked at all of the number of private variants within exomic regions within this one pedigree. And what we saw is we see at least twice, we see 45 or 46,000 variants within the exome that were seen at least twice within a pedigree, right? That's a huge number. If we look at novel, that is novel, having never been reported before in any of the bioinformatics things that we're looking at, it's about half of those. And so on average, there were 60 novel variants per founder, right? So what that's telling you is, indeed, you do get dramatic increases within of consistent common variation. So all we're really doing here is showing you another example of Mendelian mechanics. Here's an additional thing. This is actually looking at family 1 of this pedigree. And what we're really looking at is this particular man here. So this man is one of our founders. And all I did was go through the pedigree that we've studied and say, how many possible times can a private allele that he had be seen within this pedigree? And the answer is 105, right? So this isn't actually how you would do this. If you're actually formally doing this experiment, you have to take into account the likelihood that that somebody would get the allele or not, and there's much more complexity to it. I'm simply doing this to make the point that if you went out and you randomly took 1,000 individuals, you're going to have a much less likelihood of seeing multiple copies of rare events because they're rare by definition, right? But if you take people who are related to one another, you're going to increase the likelihood that you see that. Okay. So, but it's still a daunting task. So, okay, fine. If you give it to me that families are useful for this, and you give it to me that rare variants are useful, what are we going to do in order to be able to search the genome? Because we still have lots of data. So what we tend to think is that you need some level of prior hypothesis, because naive searches of the whole genome destroys any power. Now, this may be directly contradictory to the statements that JB was making earlier today about saying, well, if you constrain your data space in certain ways, you're going to reduce your potential for replication. And that may be true. I don't know. We'll have to think about it someday when I get older. But what are some strategies for reducing the total number of variants? One is that protein altering variants might be important, right? So you might want to look at variants within an exome. And among those protein altering variants, those that are predicted to be damaging might be a really good place to start. And we'll talk more about that in a minute. Then you want to think about putative regulatory variants. The only problem with regulatory variants is that, with the exception of a few particular genes, we don't know a lot about which variants are actually regulatory and which are not. Right? We're making lots of progress, and we've made lots of progress in the last few years. But I can't tell you how often I've looked at a variant that I was quite convinced was regulatory based on one particular set of experiments, only to find that no other experiment found that. And in some cases, figuring out whether something's regulatory in your own sample is the way to go, and then you get into the chromatin type experiments that we were talking about previously, or others. Then you can look at conserved variants, or variants showing evidence for natural selection, or more specifically where we started this, you can look at within QTL regions from linkage or association. Okay, so now we have some global hypotheses that will let us reduce down the total amount of space that we're seeing in the whole genome sequence data. And I told you we would think a little bit about how to define deleterious variants. So there are two major databases that people tend to use. Whoopsie. There are two databases. One of them is polyphen2, and this polyphen2 is designed to predict the impact of an amino acid substitution on the structure and function of a human protein, and it's actually done at the protein level. There's another one that's often used called SIFT, which predicts whether an amino acid substitution affects a protein function based on sequence homology and the physical properties of the amino acid. This is actually primarily computation. I have consistently utilized these two databases back and forth, but recently I went to look and said, okay, I took 800 variants that seem to be influencing brain-related traits, and I looked at the concordance between SIFT and polyphen, and what I showed was it was only 0.45. So the concordance between these two databases was really low. So this is a problem for us, and it points us to saying all bioinformatic prediction at this point really needs to be biologically confirmed in your own sample. And that's just where we are with this type of level. All right. So I'm going to give you one quick example of looking at this within humans, and it's from the Genetics of Brain Structure and Function study, which is an extension of, I don't know why I keep going the wrong way. There we go. All right. We took 890 individuals with anatomic scans. They were fairly straightforward anatomic scans. We ran them through FreeSurfer. We wanted to focus on subcortical regions. We did the genetic analyses in SOLAR, which was written by Laura Almasy and John Blangero. We looked at subcortical regions. We showed that they were all heritable. Not terribly surprising, but even in this sample they are. And then we wanted to look particularly at the putamen, because that's where Derek's big hit was in the GWAS paper that came out before. We had 118 individuals who had direct whole genome sequencing. 600 of those were from complete genomics, and 400 of them were from Illumina. And try as I might for the last year and a half, we still have problems merging these two data sets. So I'm actually only going to talk about the 600 from complete genomics. We got about 30 million variants in here. Going down, we have 87,000 or 88,000 that are non-synonymous, highly deleterious variants that are based on the polyphen2 score. Okay? Of those, 6,000 of them were variants with 5 or more alleles in our pedigrees. So I'm going to focus on those, because saying I have 5 of these rare variants allows me to run a chi-square test with a reasonable distribution. And so what happens? We come and we ran this, and we found a particular variant that was able to overcome my measure right there. That particular variant is on 6p22. It had a polyphen score of 0.99. By the way, I got lucky on this one because SIFT also said that it was highly deleterious, so okay. It's a missense mutation, and it changes a particular amino acid. In the 1,000 genomes from Mexican Americans, it had a minor allele frequency of 0.01. In my pedigree, it had a minor allele frequency of 24%. That's because we're dealing with families, and it's just an enrichment here. Important, the allele accounts for about half a standard deviation of decrease in putamen volume, right? Or if you think about it, because everything's getting smaller with age, it accounts for about 12.5 years of decreased size within the putamen. All right. What's interesting is 6p22, this particular SNP, is on the edge of the major histone compatibility region. There are 5 genome-wide significant variants within 85 kilobases of this for schizophrenia. So we're kind of interested in that. And it's in the variant influences protein created by the MOG gene, the myelin oligodendrocyte glycoprotein gene, which has been looked at a lot with regard to white matter measures. And of course, I'm starting with a gray matter volume, but there's something interesting there. Okay. I think I will actually end now, if that's good. Okay. Oh, wait, there is one last thing I have to show you, which is our conclusions and my various people that I work with, particularly John Blangiero, who was supposed to have given this talk. And here's a picture of him that an institution made next to the word power, because it just makes him seem even more of an individual of note and importance. Thank you. Questions or concerns? I will say that this particular session has been really great, and every speaker until I got up was really good, and hopefully Jason will pull us back from the brink of dismay.
No, I think you were wonderful, David. Thanks very much again. Just while Jason is setting up, I mean, I just was interested to see that your, I think Cohen's D effect size is 0.5, which is kind of in alignment with like a big effect size for rare variant. So it kind of sort of makes sense on this level.
Well, the big issue here is that if you start to do the biology past when you're doing the whole genome sequencing and you've identified a variant and you're convincing yourself that the gene is present, pretty quickly what you do is you find yourself in meetings with people who run laboratories, and you're saying to them, hey, I want you to follow up on this, right? And if you say, hey, I want you to follow up on this, and I get a Cohen's effect size of 0.001, they're going to look at you and say, there's no way I'm going to be able to see this. Mm-hmm. But if you have something at a half a standard deviation, you got a shot.
Yeah. Thanks again. That was wonderful.